Hi there, my name is Arial Wagner and I am in study group 4, and this is our second case study. I'm doing section 2, which is the forensic application of our case study. The forensic technique used in this particular case is the estimation of the postmortem interval, or called the PMI. Postmortem interval estimation is where forensic experts determine a timeline of death for a deceased individual, usually when the body is found outside or exposed to nature and weather conditions. Determining a PMI is important because it will narrow down the list of missing persons potentially linked to the remains found and how long the remains have been in that particular place. For this case, the case officials were focused on identifying who these skeletonized remains belong to. In the recent years of the remains being found, there was only one person who was reported missing from that area in Portugal. So the analysts needed to see if the remains matched both the description of the person who went missing and to see if it matched the timeline of the disappearance versus what the PMI was. The individual in question was a 61-year-old man. He was about 156 centimeters tall, which is about 5'11". He walked with a limp and he went missing 6 years prior to those remains being found. All of the initial parameters of the skeleton's biological profile and osteological analysis fit this description. However, there are no dental or clinical records for the individual, so there's no way to DNA match. So they just had to go off of what they knew from remains. These remains were found covered in botanical material, which helps because they were able to observe both the taphonomic analysis of the remains and also the progression of the botanicals found. The analysts used previous studies of late stages of composition and skeletonization of the human remains to help determine the PMI, but they ran into some problems with the botanical findings because there was not a lot of past studies or findings from that specific region in Portugal. So a lot of what they were finding were just like new plants that they hadn't discovered yet. So they don't know a lot about them. So they had to work with what they knew from other plants that they do know of. The remains found were almost completely skeletonized and almost the complete skeleton of the body. They were highly disarticulated, which means that they were most likely laid next to or on top of a shrub. And then several roots of the young shrub grew through the remains, skewing some of the remains over years of the root growth. Some of the other botanical material found on the remains were green algae and mosses covered, which covered the bones and parts of the remaining clothing pieces they found. There was multiple staining, sun bleaching or soil stains from the same fragments of the same bone indicate that they were both in direct sunlight and also under shade or under soil, something like that. Common weather decomposition is found on some of the pieces as well, that were above ground for most of their decomposition. So it just kind of like haphazardly, like there wasn't like a grave, like it wasn't like they weren't all under the soil. Because the prior analysis was so limited to the location being so remote, the PMI analysis primarily came from what they knew about botanicals found previously in other locations. The analysts took into account the growth cycles of the flora found both on top and what grew through the remains. The two types of botanicals found present were bryophytes, which are mosses, or vascular plants, which are shrubs. I'm going to share my screen real quick. Okay, so this is the rib cage that they found of these remains. As you can see, there's a lot of small roots, which means that they just, like the shrub roots, grew over time and just completely warped the rib cage of this of these remains. And this is the bones and what most of them look like. They had this like green patchy moss found on them, so they had to figure out how long these had been growing based on what they knew about other mosses. The growth period for a bryophyte can be determined either from the annual segments in the plant shoots, where there are two different growth forms. There's sympodial or monopodial. The species found on the remains were monopodial, where the apex of the plant grows longer and taller over time without breaking into multiple sections. I'm going to share my screen one more time. So on the left side, you can see this is a monopodial branching, which means that there's one stem, and then there's branching off of it. And so as it creates more branches over time, that's how old you can tell it is. The right side is a sympodial branching structure, which is where the main stem kind of breaks off into branches, and then those branches become the stems, and so on and so forth. So the remains had the monopodial side, so the left side. Those are what they found. The vascular plants found in and around the rib cage of the remains mainly were analyzed for their age using their own growth analysis, which is where they look at the growth of the root rings for the plant. However helpful this could be, there will always be a risk when trying to determine the age of rooted plants, because roots are like a plant's organs, and so they could come with their own necrosis and various issues of their own in that sense. So this could be a good technique used for gaining an estimate of the vascular plant's age, but it most likely will just be an approximation. Like I said, like they're just discovering some of these plants for the first time, so they just kind of go off of what they know of other plants. After all the analysis, it was determined that the remains did in fact belong to the man who went missing 6 years ago. Based on the PMI of the skeletonization, they said it's around 6 years old. That's when the guy went missing. The botanical growth techniques were also used to determine the age of the plants growing on and within the remains. Thank you.